Alright guys, my name's Yale and I'm here to talk about molecular motors. So a molecular motor is a protein that interacts with the cytoskeletal system and converts chemical energy in the form of ATP to mechanical energy to provide some valuable cellular functions, including active vesicle and protein transport, muscle contraction, and so cell motility, which of course is a cell's ability to move. So there are three major classes of these motor proteins. We've got the dyneins, the kinesins, and the myosins. So kinesins and dyneins walk along microtubules and carry cargo. If you think of the endoplasmic reticulum as the factory floor and the Golgi apparatus as the Amazon, then these two proteins are the UPS of the cell. So dyneins move towards the minus end of the microtubule. That is, they most often bring cargo from the outside of the cell towards the center. Now kinesins take things from the cellular center and move it towards the outside. Now dyneins are also important in cell mitosis, where they position the mitotic spindle during the cytokinesis. So myosin interacts with actin to cause contraction. Of course, actin is our microfilaments. You see in the animation here that the heads move along the actin to cause a contraction. And this is how muscle. Another important function that molecular motors provide for a cell is cell motility, which of course is its ability to move. So on the left here we have an example of how a cell would power a flagellum or a cilium. So we see here that if we add ATP to the complex here, it causes one of the microtubule doublets to slide up on the other one. And this causes the whole complex to bend to the left. Now in a flagellum, the cell would bend it to one side, to the other, back and forth, causing a beating mechanism, which is how, of course, the cell moves. Now over here on the right, we have an example of pseudopodia, where the cell rapidly polymerizes its actin filaments, or its microfilaments, to create a protrusion, i.e. a pseudopod, or false foot, that it can use to move. So some important contemporary research on the topic of molecular motors. I found that dynein in particular is very important in neurological disorders. So the first paper I looked at examines the role of dynein in Huntington's disease. Now the authors of this paper claim that dynein is regulated by a protein called huntingtin, which is mutated in Huntington's disease. So when they inhibited dynein function in brown and white adipose tissues, they found that the cells were unable to break down lipids, which is a symptom that contributes to Huntington's disease and the symptoms therein. So the second paper I looked at the role of dynein and Golgi apparatus pathology in nervous system disorders. The authors here claim that the Golgi apparatus is particularly important in forming neural tissue, and that dynein is important in maintaining the shape and function of the Golgi apparatus. So, they too inhibited dynein function in cells, and what they found is the folds and functionality of the Golgi apparatus showed some of the same problems as they've observed in actual nervous system disorders. Pretty cool stuff. Well, thanks for listening.